Hello, everyone, and welcome to this Anabios webinar. My name is Chris Mathis. I'm the Chief Commercial Officer at Anabios. I hope you've all found a nice, quiet place in your home. Those of you that are working from home these days, you're really going to enjoy this presentation. This is the first in a series of pain webinars that we have this month and next month. So be sure to register for the next two webinars. Also, we have Dr. Cheryl Stuckey on June 18th, and then Dr. Laura Stone on June 23rd. This day and age without conferences, we found that it's important to be able to keep up to speed on what's going on in the pain research field. And so we are delighted to present these webinars in collaboration with these great scientists. So before I introduce Dr. Price, we'd just like to tell you all a little bit about Anabios. I know many of you know Anabios and we collaborate with you, but to those of you that have not heard about Anabios, we are a unique contract research organization and biotech company based in San Diego, California. We procure human tissue samples from ethically consented donors and use the tissue or cells to perform physiological assays in which we can test preclinical compounds for drug discovery projects. We have essentially redefined first in human. So for more information about Anabios, feel free to reach out to me directly or check out our website at anabios.com. So it's my pleasure now to introduce Dr. Ted Price, and be sure to stay tuned at the end. We will do questions and answers at the end. Dr. Price is one of the foremost researchers in understanding how neuronal plasticity alters the way the brain receives nociceptive information from the sensory system and how this relates to chronic pain in human patients. Ted has discovered several novel targets, Ted and his team, that regulate the excitability of sensory neurons after injury and that mediate plasticity in the central nervous system that cause pain to become chronic. Dr. Price's work has been published in numerous scientific journals, and his research program has been supported by the National Institutes of Health and the Migraine Research Foundation. He serves as an editor for several leading industry journals and received the Patrick D. Wall Young Investigator Award from the International Association for the Study of Pain. Dr. Price also received the John C. Liebeskind Early Career Scholar Award from the American Pain Society, the Burmester Rising Star Award from UT Dallas, and has been named a permanent member of the NIH Somatosensory and Pain Study section. Dr. Price earned his bachelor's degree from UT Dallas and a doctoral degree in pharmacology from the University of Texas Health Science Center at San Antonio, and he did a postdoc at McGill University. So Anabios has had the pleasure of collaborating with Dr. Price and his team over the years. In fact, his 2018 paper published in Pain with his colleagues is something that I refer to quite often with partners looking for particular targets in human DRGs. For that project, he acquired some lumbar samples from normal and pain donors, and people looking for different expression of different targets can refer to that paper. So without further ado, I'd like to introduce Dr. Price, and his presentation today is Human Dorsal Root Ganglion Transcriptomics for Pain Mechanism Discovery and Therapeutic Development. Ted, take it away.
Thanks very much, Chris. I hope you can hear me. Is that true, Chris? Can you hear me okay?
Yes, thanks, Ted. You're all set.
Okay, good. So thanks very much for that very nice intro. Thanks to Anabios for inviting me to do this. And as Chris mentioned, we've worked pretty closely with Anabios over the years, and I'm going to be showing quite a bit of data from our ongoing collaboration, which has mostly been around working with human dorsal root ganglion tissue that Anabios has obtained and that we've done molecular studies on. So I do have some disclosures. Let's see. So in addition to being an academic scientist, I'm very interested in taking what we discover and turning it into therapeutics. And to that end, we started a couple different companies today. I will talk a little bit about our work on 4E Therapeutics because it's directly related to some of the topics I'll talk about in my talk today. This is our big group at UT Dallas. We recently started the Center for Advanced Pain Studies, which is supported by UTD. The center is co-led by myself and by my colleague Greg Dussor, who I've worked with for several decades now. Greg and I were both PhD students at the same time in Ken Hargreaves and Chris Flores' lab at UT Health Science Center, San Antonio, and we've run our group jointly now for quite some time. We've recently been joined by Michael Burton, who's here, who's doing some really terrific work on neuroimmune interactions as well as aging and pain. So I think you'll be seeing some really wonderful stuff coming from his lab very soon. And I'll talk today about the work of a couple of postdoctoral fellows and graduate students in my lab. I'm going to talk a little bit about the sequencing work that Pradipta Ray, who's right here, has been doing on human DRG. I'll talk quite a bit about the RNA scope in situ hybridization work that Stephanie Shires has done. Stephanie just completed her PhD with me and is now doing postdoctoral work with me. And then I'll, towards the end of the talk about some interesting computational neuroscience work that Cam Page and Andy Wang Zhu have been doing in our lab. So pain is a huge clinical problem, as I think everybody who's tuning in here knows. And we don't have very good options to treat chronic pain. This is one of my favorite paintings from one of the world's greatest artists of all time, Frida Kahlo. Frida Kahlo, in addition to being a exceptional artist, also had a pretty severe pain problem that she had for most of her life. When she was a late teenager, she was in an accident in Mexico City and had a spine injury, and she had many surgeries over the course of her lifetime to try to fix the pain that she had from this spine injury. And unfortunately, I don't think that she ever really got any kind of medical relief for her pain. I think her art was a major outlet for that, and this particular painting called Without Hope really shows the kind of anguish that she was in trying to recover from one of these surgeries. Unfortunately, even though we've had a huge amount of basic science work that's been done in the field, the situation in terms of therapeutics hasn't changed very much from the time of Frida Kahlo. I think there's a lot of hope on the horizon. We've had a lot of new therapies get approved recently, in particular CGRP therapies for migraine, and I think there's some exciting stuff on the horizon for neuropathic pain. But as a whole, I think we can all agree that we can do better. And I'm going to talk a little bit today about how we're trying to think within our group at UT Dallas and our collaborators on advancing new therapeutics, in particular for neuropathic pain. So we've spent a good amount of our time working on how inflammatory mediators and other signals from immune cells can act on nociceptors to change their excitability. And we know that changes in gene expression are really important for this, and we've spent a lot of our time focusing on translation regulation in particular. And to this end, in the last couple of years, we've published quite a few papers demonstrating that downstream of many cytokine receptors, G protein coupled receptors, and tyrosine receptor kinases, you get activation of the MAP kinase pathway. And this leads to activation of a kinase that's downstream of ERK that doesn't get a lot of attention actually in the neuroscience field. This kinase is called MINC. And this kinase is actually very specific for phosphorylating a particular protein called EIF4E that binds to the 5' end, the 5' cap of mRNAs, and regulates the translation of a very specific subset of mRNAs that in nociceptors at least seem to play a really important role in excitability. So we're trying to tease apart exactly what those mRNAs are and how they work. And we've got a couple papers on the horizon related to that, but we're pretty convinced that MINC is a potential really good target for the treatment of neuropathic pain. So why do we think that? Well, we've done many experiments in animals using behavioral approaches, behavioral genetics, and also pharmacology. We also have done lots and lots of electrophysiology. And I like this experiment because I think it kind of wraps up the whole picture very nicely. So this is a set of mouse dorsal root ganglion neurons that are put onto a micro electrode array. So there's a bunch of electrodes here that can record the activity of these neurons. And what we do is we record the baseline activity in wild type mice and also in mice that lack this phosphorylation site for MINC on EIF4E. At baseline, there really is no activity in the dish. But if we add IL-6, you see in this raster plot, so each line here is an action potential, you see that the activity of the neurons fundamentally changes. They become very excitable. They start firing lots of action potentials. And the key, I think, really is when you take the IL-6 away, these neurons have a changed phenotype. So now they continue to fire action potentials even though you've taken the stimulus away. So this suggests to me that this relatively brief exposure to IL-6 can cause these neurons to transition from being inactive to being spontaneously active. And I think that many of us in the pain field think that spontaneous activity in nociceptors is a key driver of neuropathic pain. So what happens in neurons taken from mice that lack this EIF4E phosphorylation site for MINC? Well, they show an initial response to IL-6, but it pretty rapidly tails off and it never reaches the magnitude of what you see in the wild type. But the really important thing, I think, is that when you look at later time points, these neurons don't ever really transition to having spontaneous activity. So taking out MINC signaling seems to block the development of spontaneous activity in sensory neurons. So those experiments were done in mice, and that's actually the only experiment, except two experiments at the end that I'll show you that's mouse data. The entire rest of the talk, I'm going to talk about our work in human dorsal root ganglion. So as I mentioned at the start, we've had a really great collaboration with Anabios, but we also have another source for dorsal root ganglion tissue, and that's through our collaboration with Pat Doherty and his colleagues at MD Anderson. So at MD Anderson, they have patients that have cancer, and some of them have cancer that's infiltrated their vertebrae. So you can see here, there's a large tumor. This patient is at risk for major spine injury because of this tumor. And the patient is going to have a surgery called a vertebrectomy, where they go in and they take out the tumor and the surrounding vertebrae and basically rebuild the spine around the spinal cord. As part of the process of doing this surgery, the dorsal root ganglia are often in the way and are actually removed. So this creates a really great opportunity for us because we can phenotype these patients beforehand. So for instance, in this particular patient, we know that they have pain in certain dermatomes. This particular one has pain on one side of their body and not the other one. So when we take out these dorsal root ganglia, we can then do electrophysiology and RNA sequencing, which is what we have been doing over the last couple of years through this collaboration, and get an idea of what is changing in DRGs that innervate painful areas versus DRGs that are potentially normal and are not causing neuropathic pain in these patients. So one of the first things I mentioned was that the spontaneous activity seems to be a key driver of neuropathic pain, but we don't really know if spontaneous activity is present coming from the DRG cell bodies in patients with neuropathic pain versus patients without neuropathic pain. So this is one of the first experiments that we did along these lines, and all this electrophysiology was done in Pat's lab by Rob North and Yan Li. And essentially what they found is that it was extremely rare to see spontaneous activity in DRG neurons taken from patients that did not have pain. But if we looked at the DRG neurons that came from the dermatomes where they did have pain, about 20% of those neurons showed spontaneous activity in the dish. So this is, I think, good evidence that neuropathic pain is accompanied by spontaneous activity emanating from the soma of DRG neurons. So this provides an opportunity then to test hypotheses that we have that previously we could only really look at in animal models. So we, as I mentioned before, are very interested in MINC and EIF rephosphorylation as a driver of spontaneous activity in neuropathic pain. We have lots of animal model evidence for this, but we can use a MINC inhibitor. In this case, we use an inhibitor called EFT508, which was developed by a company called Therapeutics. And if we put this inhibitor on a DRG neuron that shows spontaneous activity, you see that this is reversed, suggesting that blocking MINC activity can eliminate spontaneous activity in human nociceptors. So we think this is a really good kind of clinical bridge to take this idea forward and try to develop a therapeutic. So we've done exactly that. We started a company about a year ago called 4E Therapeutics. We have recruited a CEO, Craig Benson, who's at the head of this company. And then Jim Son is our head of drug discovery. He's a medicinal chemist. He's also in Austin. So our company is based in Austin. And a little less than a year ago, we received a large phase 1 U grant from the HEAL initiative from NIH to advance our ideas around developing MINC inhibitors for neuropathic pain. So the goal of this project is to optimize lead drug compounds and then eventually prepare an IND application to move towards phase 1 clinical trials for neuropathic pain for MINC inhibitors. So we've made a tremendous amount of progress on this actually over this relatively short time course. We've now identified many new MINC inhibitors. We also would like to eliminate brain exposure for these MINC inhibitors because we're concerned about potential CNS side effects of blocking MINC over a long period of time in neuropathic pain patients, and we've also been successful in doing this. And now we have a series of very potent, very specific MINC inhibitors with minimal brain exposure, quite good oral bioavailability, and we're now optimizing their drug-like properties to eventually do in vivo efficacy tests and also screening on human DRG neurons in collaboration, again, with Pat Doherty's lab. With the goal of identifying a lead compound and starting our IND process next year. So we're very excited about that. So that summarizes kind of what we've been working on in terms of electrophysiology and also drug discovery for targets that we gleaned from animal models. Now I'm going to transition to talking about the molecular work we've been doing on human DRGs to try to identify new targets for neuropathic pain that are found completely independently of animal models. So on all these dorsal root ganglion we've been getting from Pat at MD Anderson, we've been doing RNA sequencing. We've now sequenced about 60 DRGs from these patients. So this looks like a little bit of a busy slide, but it's not. It's color coded, and I'm going to explain how to look at it. And I'll make a point about what we think might be some interesting drivers of neuropathic pain in males and females in humans with neuropathic pain. So all of the yellow genes here are things that were upregulated in males with pain versus females. And all of the purple genes are things that were upregulated in the females versus the males. And then all of the genes that have these little green diamonds on them are genes that are enriched in macrophages. It doesn't mean that they're only found in macrophages, but they're relatively enriched in macrophages. And then genes that have these red stars on them are enriched in neurons. So hopefully now that you know the color code of this, you can see that the yellow stuff is very heavily weighted over here, and it's very macrophage heavy. So we think that this is a good indication that, consistent with the preclinical literature, there is a very strong macrophage drive for neuropathic pain in males. But in females, this picture looks quite different, and this is also actually consistent with the emerging preclinical literature. So there's been a lot of work now done showing that microglial contribution is kind of hard to pin down, and macrophage contribution within the DRG is kind of hard to pin down in female rodents with neuropathic pain. What we see is that many of the genes that are upregulated in females are G protein coupled receptors. And we're pretty interested in the idea that many of the changes that are happening in neuropathic pain in the female DRG are actually intrinsic changes in gene expression within the neurons, rather than being driven necessarily by a particular type of immune cell. So to try to get into that in a little bit more detail, Pradipta Ray has been working on trying to look at specific mechanisms that might be driving neuropathic pain in particular individuals. So one of the things that Pradipta noticed when we first got started on this idea was that we had a relatively small subset of patients that had really, really strong upregulation of BDNF. So I think most people in the pain field are familiar with BDNF. It's a neurotransmitter that acts or it's released by sensory neurons, and it can act on dorsal horn neurons to cause synaptic plasticity, like long-term potentiation. So it seems to be a key factor that's released from the peripheral nervous system that causes central sensitization, and it's upregulated in many preclinical pain models. And we saw that there was really strong BDNF expression only in a relative small subset of patients from the study with MD Anderson. What was really interesting is that when Pradipta did a analysis that looked for co-regulation of other genes with BDNF, he identified Ganglion, which is another neuropeptide, which is also known to be upregulated in preclinical neuropathic pain models. So this suggests that there might be different mechanisms that drive neuropathic pain in different subsets of patients. So we decided to look at this a little bit more broadly. And now Pradipta has identified 4 subsets of patients that we think might have different mechanistic underlying drivers of their neuropathic pain. So first we have this subset that's a mix of males and females that has a really strong upregulation of BDNF and Ganglion. These also happen to have strong upregulation of GFR alpha-1, which is a GDNF receptor that people like Mike Jankowski in the pain field have identified as another particular contributor to nociceptor hyperexcitability. We also identified another subset that has strong TRPM8 upregulation, as well as TrkA receptors. So we think that this particular patient population, for instance, might have strong cold hypersensitivity and might respond to anti-NGF therapeutics. We also found a larger group, which you might have guessed from the previous slide, that contains a bunch of genes that are relatively enriched in macrophages. Of course, none of these are actually macrophage-specific genes, but they're found in macrophages. That's like one thing that they all have in common. These include a bunch of transcription factors, interleukin-6. Interestingly, almost all of these are males. So we think this is our group that has this underlying macrophage infiltration or proliferation that might be driving neuropathic pain. Finally, we have a group that has another set of immune genes. There are quite a few toll-like receptors, as well as chemokine receptors and the P2Y13 receptor. We don't know exactly what immune cell these are coming from, but one thing they have in common is that they're all expressed by B cells, so that's a hypothesis that we're interested in. And then another thing that's very interesting here is that these are mostly all females. So potential different drivers, macrophages and maybe B cells, a hypothesis we're exploring in females with neuropathic pain. So all that data I just told you about is bulk RNA sequencing. We think bulk RNA sequencing is useful because we can look at lots of different cell types at the same time, but ultimately we really need to start to figure out at the cellular basis what's happening here. So, you know, that example here of this subset is a great one for that. We think it could be B cells, but we really don't know at this point. So we need to get down to better cellular resolution. So to that end, we're doing lots of RNAscope and immunohistochemistry on human DRG samples. This particular sample here is from a peripheral nerve from a diabetic patient that had a lower leg amputation. So we're very interested in looking at local changes within nerves of people with neuropathies. So Stephanie has really been pushing this ahead in the lab and has done more RNAscope than I ever thought possible on human DRG samples. And I'm going to just show you a little bit of data about how some of the things that we think we know about the DRG from mouse studies. We may know some generalities, but there probably are different populations of sensory neurons in the human DRG than there are in mice. So I'm going to give you some specifics on that. So the first thing that we tried was to look at the so-called peptidergic versus non-peptidergic population. So you can't really do IB4 staining on human DRG because it doesn't work. IB4 doesn't stain anything in the human DRG. So what we did instead was to use CGRP, calc-A gene, and P2X3, the ATP receptor that's found in these IB4 positive neurons as markers for these populations. So what we see in the mouse is very consistent with a very long literature where these populations are mostly segregated. There is a little bit more overlap at the mRNA level than what you see at the protein level between these and mouse, and this is actually very consistent with some single cell sequencing experiments coming from the Lenarson and Ernfor's labs. But in human, there's far more overlap between these, and that mostly happens because there's an expansion of the total percentage of neurons that express the CGRP gene. So in human DRG, about 60% of the neurons seem to express calc-A mRNA, whereas in mice that it's closer to 30%. So there's far more overlap between these populations in humans than there are in mice. So another key gene that we looked at was TRPV1. So TRPV1 is mostly found in the peptidergic population in mice, which is reflected here. But we were pretty surprised in human DRG, and we've done this now from 3 independent organ donors. Essentially all of the smaller diameter neurons that express these nociceptor markers also express TRPV1. So it seems like in human, the vast majority of putative nociceptors likely express TRPV1. So there's an expansion of the CGRP expressing population, expansion of the TRPV1 expressing population, but the TRPA1 population is decreased. So in mice, TRPA1 is mostly expressed in the IB4 population, and then there's a shrinking of this TRPA1 positive population in humans. So there's some interesting changes in these populations that we've all kind of grown to know from mouse DRG. There are also some striking examples of genes that particularly for receptors that are not expressed in the mouse DRG at all, but are actually found in human DRG. So this is one of my favorite, the alpha-9 nicotinic receptor. There's been a lot of really interesting group work done from a group at the University of Utah looking at alpha-9 nicotinic antagonists as potential therapeutics for neuropathic pain. And alpha-9 is expressed by T cells in mice where it's thought to act to reduce neuropathic pain. But there's a striking species difference here. In humans, alpha-9 nicotinic receptors are expressed in many DRG neurons, and they seem to mostly be found in this overlapping CGRP and P2X3 expressing population. So I think that this is interesting from the perspective of thinking about alpha-9 as a target for neuropathic pain, where you might get a kind of double whammy in humans, where you could have a beneficial effect both on DRG neurons and on T cells. We also have become very interested in finding targets from our transcriptomic work, and then looking at where those receptors are found within the DRG. So one of the things that we noted in our bulk sequencing experiments was that oncostatin M, which is a cytokine, was very strongly upregulated in our neuropathic pain patients. So oncostatin M acts via a IL-6 receptor family receptor signaling complex. So it contains the GP130 signal transducer, and then the oncostatin M receptor, or OSMR. So we wanted to explore where this was expressed in the human DRG, and what we found was that many human DRG neurons, about 20% of them, express the oncostatin M receptor. This receptor also seems to be expressed in neurons in the mouse DRG, but interestingly, when we looked, we saw this very clear signal around neurons as well. So these are satellite glial cells that surround many of these DRG neurons, and these express really robustly the oncostatin M receptor. This is interesting because in the mouse DRG, from single cell sequencing experiments, which have mostly been done by the Lenarson lab, satellite glial cells do not express the oncostatin M receptor at all. So this suggests a potential expansion of signaling of oncostatin M within the DRG in humans that might be involved in neuropathic pain. So to summarize all this kind of molecular work that we've done on the human DRG so far, we see that there's this expansion of the CGRP population, and there's a great deal of overlap between calc-A and P2X3 compared to mice. And additionally, there are far more human neurons that express TRPV1, suggesting that probably most human nociceptors at least express the mRNA for TRPV1. So we probably will not have the luxury of taking DRGs out of neuropathic pain, out of your average everyday neuropathic pain patient, to do this kind of molecular profiling and try to understand what's driving their neuropathic pain anytime soon. But we can sometimes obtain nerves from these patients through nerve biopsies or things like that. We can also get skin biopsies from most patients. In fact, for certain types of neuropathies, that's a standard part of their clinical diagnosis. So another thing we've been focused on is trying to examine whether or not mRNAs are robustly transported in axons of these sensory neurons, and if we can find them in the peripheral nerve. If that is true, it suggests that you could get relatively small samples of the peripheral nerve and get some insight into how the transcriptome is changing within the DRG by sampling these transported mRNAs. So here, what I'm showing is TRPV1 and SCN9A, NaV1.7 mRNA found within a human sural nerve using RNAscope. And you can see that there is both TRPV1 and NaV1.7 signal. Interestingly, a lot of the signal seems to be in Schwann cells. So it somehow is making its way out of the axons, or some of these Schwann cells within the nerve actually express these mRNAs. So we're actively trying to figure this out using a variety of sequencing technologies, but I think it has some promise for being able to sample peripheral nerves. And this shows you piezo2, another sensory-specific gene, and we can also detect piezo2 mRNA in human sural nerves. So that's one way we could potentially do this. Another is we could look at interactions between diseased tissue and human nociceptors. So that's what Andy and Candler have been focusing on in their work. So the idea here is that we have a good baseline data set for what receptors are expressed in the human DRG. And we could take samples from individual patients, for instance, synovial fluid, do sequencing, compare the ligands that are found in their cells in their synovial fluid versus the DRG neurons, and identify ligand receptor interactions that might be important for driving pain in that individual, and then tailor therapeutics around that. So we had to do a lot of kind of baseline work to make this work. So Andy and Candler really kind of poured themselves, as well as many of our undergrads on our team that worked on this project, into curating lists of ligand receptor interactions. And we finally developed a list of about 3,000 interactions between ligands and receptors. And then Andy built a platform where you can take RNA sequencing experiments from different sources and run these against each other to find the ligand receptor interactions within those particular data sets. So I'll give you just a couple of quick examples of how we've done that. So one relatively straightforward thing that we could do is to use some of these single cell sequencing data sets from the Linnarsson lab in Sweden and separate out different classes of sensory neurons, as well as the support cells, the satellite glial cells and the Schwann cells that are found within the DRG, with the idea that we can look at how intraganglionic transmission from neurons to these glial cells and vice versa might influence each other. So this particular one is looking at neuronal ligands versus glial receptors, so receptors that are expressed by satellite glial cells and Schwann cells. And we were really surprised here. I mean, we have not seen very many things in the literature looking at these kind of interactions, but we found some pretty robust things. One of the things that we were really quite interested in is CGRP, which of course I'm not going to be able to find on the list here right now, but CGRP is here. And CGRP receptors are actually pretty robustly expressed by many of these satellite glial cells and Schwann cells. So, one of the most robustly expressed and released neuropeptides within the DRG might be having a trophic action on these satellite glial cells and Schwann cells. So if you look at this vice versa, so now going from glial ligands to neuronal receptors, this actually expands pretty massively. It really is impressive how many ligands for neuronal receptors are expressed by satellite glial cells and Schwann cells. And I know there's a number of people in the pain field that are actively working on, especially satellite glial cells and their potential role in neuropathic pain and chronic inflammatory pain. And I think, from this kind of analysis, you can see just how interesting those particular lines of work could be moving ahead. So we wanted to try to apply this to a specific clinical situation, and there's just an absolute explosion in the number of experiments that are being done in disease states using RNA sequencing. So this particular paper was published pretty recently in Science Translational Medicine, and what these investigators were doing is that they were taking macrophages from people with either rheumatoid arthritis or osteoarthritis and characterizing those macrophages. And they identified specific macrophages that seem to be involved in disease in rheumatoid arthritis, as well as specific macrophages that seem to be involved in disease in osteoarthritis. So what we did was then to build interactomes with these from the human DRG data that we already had. And I'm just going to focus on what we found for the rheumatoid arthritis. So here, the results were, I think, pretty interesting. First, we saw a downregulation of IL-10. So IL-10 seems to be a really important factor for pain resolution, and this seems to be downregulated. So you might be losing a pain resolution mechanism. And then we also found several things that were really strongly expressed in these macrophages promoting rheumatoid arthritis, including epiregulin, which is an EGFR receptor agonist. So a really nice study from Luda Diachenko, Jeff Mogul, and Lauren Martin, published a couple of years ago in JCI, demonstrated the potential role of epiregulin in promoting many types of pain through the EGFR receptor. And we also found another ligand, HBEGF, that also acts via the EGFR receptor that was pretty strongly increased in rheumatoid arthritis patient macrophages. Oncostatin M was also found here, as well as some other factors acting on TNF receptors. And of course, TNF receptor or TNF sequestering therapeutics are kind of the standard of care now for treatment of rheumatoid arthritis. But there's a pretty complex mixture of potential pain promoting agonists that are expressed here in these rheumatoid arthritis macrophages. So we decided to focus in more on HBEGF because essentially nothing is known about HBEGF in the context of pain. So having this human data in hand, we decided to go back to the mouse, and we did two experiments. We first tested whether HBEGF could actually activate mouse sensory neurons. So here we use calcium imaging to do this. And we saw that about 30% of mouse sensory neurons respond to HBEGF, and that HBEGF effect is completely blocked by lapatinib, which is a EGFR receptor antagonist. So HBEGF does seem to be able to excite mouse nociceptors, and it does it via the EGFR receptor. And then we tested whether or not this EGFR agonist can cause mechanical pain or grimacing in mice with injection into the hind paw. So what we saw here is that in both males and females, we get mechanical hypersensitivity. We get grimacing, which is a little bit more robust in the females than it is in the males. And if you look at both of these combined, there's really nice mechanical hypersensitivity induced by HBEGF, as well as transient grimacing after the injection. So this suggests that HBEGF, in addition to epiregulin, is an EGFR agonist that can stimulate pain behaviors in mice, and it's associated with rheumatoid arthritis in humans. All right, so we're all at home because of COVID-19. So I want to just show you one more application of this interactome technology that we're interested in. So your airway is innervated by nociceptors, and your upper airway is mostly innervated by the vagus nerve. Your lungs are innervated by a mix of nociceptors that come from the thoracic DRG and from the vagus nerve. And the lungs are a key battleground in this disease, as I think everybody is very well aware now. So people like Isaac Chu have shown really nicely that nociceptor innervation of the airway plays a key role in the disease response. And we have sequenced many thoracic DRGs from the collaboration with Pat. So we decided to use that data and look at ligands coming from immune cells in the lung of COVID-19 patients. So these were from experiments that were done in China in some of the earliest severe COVID-19 patients. We used their sequencing data with a lot of help from those investigators, so they really played a huge role in helping us understand very quickly their data sets. And then we looked for interactions between those immune cells in the severe patients and thoracic DRG neurons. And so I'm not going to show you all the details. I'll just summarize basically what we found. So first of all, we found that a bunch of ligands that are upregulated in the lung of these COVID-19 patients are genes that are very well known to be regulated at the level of translation by MINC-driven EIF rephosphorylation. So we think that this could be another potential application of MINC inhibitors. So for potential alleviation of the severe lung inflammation that happens in these patients. We also saw some pretty strong indications of ligands that could act directly on nociceptors coming from the thoracic DRGs that might be promoting inflammation in the lung. So these included CCL2, which can act on a number of receptors that are expressed by human DRG neurons, epiregulin, which I was just talking about, that acts via EGFR, and then some fibroblast growth factors that have recently been described as interacting with voltage-gated sodium channels like NaV1.6. A final question is, could the virus itself potentially interact with sensory neurons through ACE2 expression in sensory neurons? So that's the next question that we set out to answer with our human DRG. So Stephanie got her hands on some RNA-scope probes for ACE2 and looked at ACE2 expression in both lumbar and thoracic DRGs from either these organ donors or surgery patients. And what we see is that ACE2 is indeed expressed in a relatively small proportion. So it's about 20 to 25% of the neurons here. And many of these neurons are positive for both CGRP and P2X3. Many of them also happen to express MRG, PRD, and NPPB. So I think this is important because we know that these MRGD positive neurons form free nerve endings in the skin and also in some of the luminal organs. So it suggests that the virus might have access potentially to sensory neurons via ACE2 expression, and that the sensory neurons that do express ACE2 are likely ones that have free nerve endings that are out at the surfaces of your skin and also your internal organs that might make them susceptible to infection by the virus. So I want to just mention one final thing related to this is that it's recently been published that in addition to loss of sense of taste and smell, that many people that have COVID-19 lose chemical sensation in their oral cavity. So that includes losing sensation for menthol and capsaicin. So it's possible that part of the way that that happens is that the virus infects the sensory neurons that are leading to those sensations and is somehow silencing them. So that's obviously just a hypothesis, but that's something that is suggested by the expression of ACE2 in these neurons. All right, so to wrap up, I just want to kind of point out where we're trying to go with all this. So first, I think we're really heavily invested in trying to create really rich resources that everybody can use. So I hope that most of you already know that we have these websites that we house here at UT Dallas, where you can go and look through any of our data sets to look at your favorite targets. We're currently trying to expand that right now. So if you search for our Sensoryomics website, you'll see that we revamped our front page, and we're trying to make all this data kind of more integrated and more accessible to anybody that is interested in it. And we'll soon have peripheral nerve transcriptomes included in there. So we're really trying to do the best that we can to make this as available as possible to everybody within the field. We're actively working on doing single cell sequencing in human DRG. We're actually doing spatial transcriptomics using the Visium system from 10x Genomics. And we're also working on doing this from nerve samples with the hopes that we'll be able to more thoroughly understand the neuroimmune component that is driving neuropathic pain in neuropathic pain patients. We are actively developing this interactome, as I just mentioned, as a way to identify new therapeutic targets. And for instance, HBEGF interacting with EGFR is one such example. And our ultimate goal is to be able to use these technologies to develop patient-specific mechanistic insight into what causes neuropathic pain. And because we are entrepreneurial, here we're getting close to launching this new company called Dolaromics, whose mission will be to try to implement this in a way that it would be accessible to investigators and also to patients. So I want to stop here and just again point out the really great people that I work with that did the work I talked to you about today, as well as all the rest of my group. Pradipta did all of the RNA sequencing analysis. Stephanie did all the RNA scope work. Andy and Candler have done all the work on the interactome. Mo did the HBGF behavior, and DJ did the calcium signaling that I talked to you about. Mo and DJ are brand new PhD students in the lab. All this work is a collaborative effort with lots of really great investigators. Greg and I have worked together again for a really long time now. Michael Burton and Zach Campbell at UTD have just been terrific to work with, as well as Taehoon Kim and Joe Pancrazio. And then our collaboration with Pat Doherty, as well as Kobe and Anamika at MD Anderson has just been terrific. Finally, none of this is possible at all without the support of NIH. And some of the work I talked about today was also supported by Merck. So I'll stop there, and I'm happy to take any questions.
Thanks a lot, Ted, for the fantastic presentation. And we do have time for questions. You can post them in the questions tab, and I will read them to Ted. You can also raise your hand if you'd like to ask the question without writing it in. I'll start while people put their questions together. Ted, it strikes me just how much variability there is to address it, and the complications, male versus female, the state of the immune system, the type of neuropathic pain, and the region of the body affected. How do you suggest that, especially those in biotech or pharmaceutical industry, go about this for developing medicines? Are we headed towards personalized medicine for the treatment of pain? What are your thoughts on that?
Yeah, so I think we should be thinking along those lines, but I don't think that it ultimately is going to boil down to every patient has some kind of different mechanistic driver. I think what we're seeing is some very clear trends of specific kind of subsets of neuropathic pain. So I'm struck by how kind of consistent the macrophage type signature is within many of our male neuropathic pain patients.
Mm-hmm.
And also how consistent that is with a lot of the preclinical literature. So I think that there are a lot of really good opportunities there. I think in the story in females, like we're tremendously behind on the preclinical part of this and understanding what's actually driving neuropathic pain in females. So there's a lot of work to be done there. But I'm pretty excited about it's not perfect consistency, but the relative consistency of what we seem to be seeing in the female neuropathic pain patients. And I think that there's a huge opportunity there because if we better understand that, it should lead to all kinds of brand new therapeutic opportunities that maybe we haven't been thinking about very much collectively. I also think that for diseases like rheumatoid arthritis and osteoarthritis, that there's great opportunities in using these kind of data sets that we can generate now to find mixes of mediators that are likely interacting with nociceptors to make them hyperexcitable in those specific deep disease states. So, I imagine that there will be a fair amount of commonalities and differences, but I think if we can understand how these factors are signaling, that there's likely to be a lot of commonality because they do seem to fall within specific subclasses of receptors, and these receptors all have conserved signaling mechanisms that they don't all signal individually differently. So I think
Right.
We can use the information and the variability to think about how we would try to approach it therapeutically.
Okay, thanks. Very helpful. So we do have several questions from the audience. Here's one. from one of your earlier slides, what happens after 24, 48 hours or a week of any change in the pattern that you observed in first 2 hours of exposure to IL-6?
So we haven't really done experiments like that. I think we have recorded for longer periods of time, and the spontaneous activity continues, but we haven't tried to examine yet whether there are different mechanisms involved in the initiation versus the maintenance, and if that changes over a period of time. For instance, if it's different at like 3 hours versus 48 hours, which would be kind of akin to your early versus late LTP mechanisms, which are quite distinct from each other. So it's a really, I think that's an extraordinarily interesting area to do work, but we have not tried to address that.
Okay. We do have a direct question from Sandy Eldridge in the audience. I'll unmute Sandy. Go ahead.
Oh, hi. Thank you very much. I assume you can hear me.
Yep, I can hear you.
Oh, that was a fantastic seminar. Thank you very much for making my day.
Sure.
So I am here from the National Cancer Institute, and our investigative toxicology lab is specifically interested in developing a model to recapitulate chemotherapy-induced peripheral neuropathy. And so, as you can imagine, it's challenging, and I'm wondering how translatable your efforts would be to CIPN, so chemotherapy peripheral neuropathy. So that's one question. I probably have a whole bunch, but I won't try to ask them all of them. So the other thing is we're concerned that maybe we think that we need to be focusing on DRG.
Yeah.
Because we think that, of course, it seemed the dogma is that that's the target for chemotherapy-induced peripheral neuropathy. But we're also wondering if maybe there's an important component between the CNS and the PNS that could be playing a role there. So, we're looking at human-induced pluripotent stem cell-derived DRG as a starter for a cell in vitro model, but also based on some of the things you've said, I'm wondering if maybe we should be looking at a mixed cell population, realizing the importance of other cell types. So why don't I stop there and let you talk? Thank you very much.
Yeah, thank you. So those are great questions, and I could talk for probably quite some time about them, but I'm sure I couldn't actually give you an answer, because I think these are things that lots of people are currently thinking and trying to work out, but I doubt that anybody feels like they really have a firm grasp on them. But so Pat and I have talked quite a bit about these patients that we get from MD Anderson, where we're trying to understand what exactly is causing the spontaneous activity. And they're really complex patients. That they have cancer. Most of them have had some kind of chemotherapy. Many of them are on pain medications, and it's hard to know why they have spontaneous activity. It's even hard to understand in some cases why some of them don't have pain, because if you look at their scans, you'd be like, this patient has to be in pain, but they're not. And the pain versus non-pain correlates extremely well with the spontaneous activity. So I know that there are groups that are working on models where you expose the DRG neurons directly to the chemotherapeutic agent, but I think there also is a really important neuroimmune interaction that's happening there, and I guess what we would like to be able to do eventually is to do enough of these kind of sequencing experiments that we're doing, and we're funded to do it now, so we will be working on it over the next couple of years, where we eventually have enough data, enough sequencing data, enough patient information where we can start to make calls about what is causing what in terms of what we see. And I think that that would be very informative for thinking about how to do the kind of experiments maybe that you're talking about. I think that the iPSC nociceptor idea is a really good one, and I know that there are lots of people that are working on this, and I think it's a really important part of our field. So I encourage it, but we haven't worked with it very much, mostly because we are kind of in this unique position where we do have a lot of access to human DRG, and we're kind of focused as much as we can be there. Although we've recently started working with Angelika Lampert in Germany to try to do some more thorough comparisons between the iPSC nociceptors that she develops versus our human DRG data. So to try to get an idea of, exactly what types of sensory neurons are there. I know there are other people like Alex Chesler at NIH that are actively working on that as well. So there's lots of really interesting work to do.
Ted, I can also comment on her question about the DRGs versus other parts of the nervous system. At Antibios, we're collaborating with Eli and Lilly to develop a human spinal cord electrophysiology assay. So stay tuned for that. So Ted, we are at the top of the hour. Are you able to stay on? We have several questions.
Yeah.
If you can stay on for a few more minutes. Okay.
Yeah, absolutely.
So someone asked about the chemo agent, I presume, in the collaboration with Pat Doherty. Was it oxaliplatin? What can you comment on that?
I mean, it varies actually. So these patients have different cancers and have been on different types of chemotherapeutic agents. Pat could probably immediately roll off which ones they were. I can't off the top of my head, but it's not a single one.
Okay. Here's a question. Do you think that a few minutes of EFT 508 treatment is sufficient to reverse translational changes that drive hyperexcitability?
It seems to, well, it depends on what your definition of few is. So it does not have an immediate effect. So I think we're all very confident that it's not just blocking a sodium channel or something like that. It's surprising how quickly it does work, though. So we're pretty actively trying to understand why it works so quickly. MINC has been studied pretty extensively, and it has very few targets other than EIF4E. So, and all of these effects are recapitulated in the EIF4E phosphomutant mice. So we also have done very extensive testing of EFT 508 specificity, as has effector therapeutics. And, this is a really remarkably selective kinase inhibitor. And so MINC has a slightly atypical ATP binding pocket, which allows for some more distinct pharmacology than what you typically get with these kind of kinase inhibitors. So that's a big advantage to making specific blockers of the kinase. So we have data that it decreases T-type calcium channels, and that it can do that relatively rapidly. It doesn't do it by simply blocking them. But I can't totally rule out the possibility that it might act on some unknown target. But it has such a profound effect on excitability that at the end of the day, whatever that target is, I guess that doesn't really matter as long as it does what you really want it to do and it's safe. And I think we know now from the experience of Effector as well as Ohm Therapeutics, which is also developing these inhibitors for cancer, that they are safe.
Here's a related question. Can you comment on the effect of the MINC inhibitor on DRG action potential activity? It seems quite fast for a translation mechanism, in particular the return of activity upon washout within 5 minutes. Do you have any independent confirmation that the phosphorylation event of the substrate occurs in that time frame?
Yes, so we do know that the phosphorylation events occur in that time frame. We do not know definitively that it's which mRNAs are being translated. So we're now using trap technology in mouse, because we can't do it in humans, to identify the exact targets, mRNA targets of EIF4E phosphorylation in mouse nociceptors. We have some clues. We're pretty sure it's not translation of the channels themselves. That doesn't make sense for the time frame. We think it probably has to do with trafficking. And again, we can't totally rule out the possibility that it is an event that is not translation regulation on those short timescales.
Interesting. Here's another verbal question from the audience. Anil Kalia, if you're still there, go ahead.
Yeah, thank you very much for a very interesting talk. I have a question that Ming signaling L4E inhibitors, what concentration is enough for the inhibition? I mean to say, how is the potency? And what about the central nervous system penetration of these compounds? And secondly, sure. What about the expression of NAV 1.7 on Schwann cells? So you was talking about Schwann cells. Do you mean to say that they are highly expressed there?
Okay, yeah. So first question for EFT508. So these are EFT508 is a really potent inhibitor. It's IC50 against the recombinant kinase is about 1 nanomolar, and its IC50 in cells is between 1 and 10 nanomolar. The experiments that we use, we did it with 25 nanomolar. So, and at that concentration, it doesn't really have any other known targets within the human kinome. We've screened against GPCRs at the PSPP at University of North Carolina and didn't find activity at those concentrations either. So we think it's a specific effect. The second question, what was the second question again?
Regarding the Schwann cells?
Yeah. Okay, so we see both TRPV1 and NAV 1.8 mRNA puncta in what appear to be Schwann cells, but I don't think that the Schwann cells really express these mRNAs. So there's have now been a couple of different papers showing that Schwann cells can form these invaginations with axons in peripheral nerves. And then, of course, there's this other paper from the Ernfors lab that was published in either Science or Nature, I don't remember, about a year ago, showing that there's direct communication at axon terminals between Schwann cells and nerve endings. So it seems to be possible that there would be some transfer of material between these cells, and it's been shown experimentally. And then one of the long-standing things in the neuroscience field that has led people to this conclusion that I don't think they really have anymore, that translation doesn't happen within axons, is that there are relatively few ribosomes in axons. But of course, Schwann cells have a lot of ribosomes, so I think it's possible that there's certain types of Schwann cells that form these invaginations where mRNAs that are transported in the axons can enter those cells, be translated, and then the protein can be transferred back into the axon. So we're trying now to test doing nuc-seq from human nerves to see if TRPV1, NAV 1.7, etc. are being actively transcribed in Schwann cells in the nerve, and I'd be fairly surprised if they were. So I think that we have a long way to go to show that that's actually what is happening, but that's the idea.
Okay, thanks, Ted. Here's another question from Angelica Lampert. She said, you mentioned FGF13 and immune cells of COVID-19 patients. When it interacts with sodium channels, it does so from the intracellular side. How does it get there? Or is it upregulated in nociceptors?
So it's coming from immune cells, so it would have to be taken up if it was going to have some action on the on sodium channels and nociceptive endings. So we're obviously not able to test that hypothesis directly in the human lung. It's also possible it could have completely different actions that are independent of the sodium channels, because there are other receptors that are also expressed by nociceptors for these FGF proteins.
Okay, and here's a question from Cheryl Stuckey, who will be our next webinar speaker in June. Given the extensive expression of TRPV1 in human DRGs, do you think efforts should be enhanced in finding pain therapeutics for this target, therapies that lack the thermoregulatory issues?
Yeah, so I guess Johnson & Johnson is now in phase 3 with their TRPV1 antagonist that has relatively smaller hyperthermia liabilities than other clinical candidate compounds, and we're going to see. So I think that's that's one conclusion you could reach, and Johnson & Johnson has continued to put their money behind this. So I guess we're going to find out.
Still an important target, yeah. Here's a question: any update on expected timelines for a single-cell human DRG RNA-seq data set?
Well, you know, I mean, I think it's probably been accomplished by at least 1 or 2 groups out there using a nuke-seq approach, and when it comes out, I'm sure it will be extremely useful to people. We've decided to do it a little differently. We're going to do spatial transcriptomics using one of the 10x spatial transcriptomics techniques called Visium, and we're starting those experiments now. So we will, I think, be generating a data set that will be a little bit different from the approach that others have probably taken. And I would imagine that the combination of these things is going to be extraordinarily useful to people.
Absolutely. Since you see commonalities between neuropathic pain models, do you think that the RNA changes you find are caused by the spontaneous activity, or are there activity-dependent mechanisms?
Yeah, that's a great question. There's really not an easy way for us to know that, although what Pat's lab is starting to do is to do patch-seq on these neurons, and I assume, you know, we could maybe eventually start to be able to answer that kind of question using that kind of technique. But we've got a long way to go before we'd be there. But yeah, that's a fascinating question. There must be some set of genes that are responsible for, you know, the origination of the spontaneous activity, and presumably they are also responsible for these oscillations in the resting membrane potential, because those two things seem to coincide, as Pat and as Marshall DeVore showed, you know, over many years, and then as Pat and Terry Walters have kind of dissected very nicely over in Houston. But then, yeah, there probably are another subset of genes that are being regulated in an activity-dependent fashion by the spontaneous activity. And I guess the big question would be, how long do you have to turn off the spontaneous activity to make that activity-dependent transcriptional change or translational change go away? And we don't have any idea.
Okay, let's do one more. Thanks for indulging us and staying on a little bit longer.
Yeah, sure.
In addition to different distribution of DRG cell populations in human DRG versus mouse, are there entirely distinct populations of cells in human that aren't seen in the mouse data?
Yeah, I feel pretty confident about that, but we're trying to nail that down, and so I'm not going to speculate too much on it, but yeah, I think so.
Okay. Well, fantastic. There are so many questions. We could go for another hour. So what I'll do is I'll send you the list of questions, Ted, and encourage those in the audience to reach out to you by email.
Okay.
Thanks again for a great presentation, especially the end about COVID-19. That was fantastic. I'd like to thank my colleague Gary Watkins, Director of Marketing at Anabios, for putting this together. And thank you all for attending. And we look forward to having you back in June for 2 more great pain webinars. Thanks, everyone.
I'm looking forward to those too. Thank you.
Have a great day, great evening. Goodbye, everyone.